Today, I'm going to share with you how to make your image analysis workflows more efficient with Imars. I will first discuss some of the prerequisites for image analysis automation. This is important because a lot of times having good quality data really is the key for smooth and reliable image analysis down the line. Then I will focus on tools in Imars that can help you to batch process and streamline the workflow. We will cover file converter, batch image processing, batch object detection, and statistics export. Hopefully, these tools will make your life easier when you are dealing with your many image data sets. Overall, our goal is to provide a user-friendly pipeline that takes the raw image from the microscope all the way to the final quantitative analysis that provides biological insights. We'll do a few demos in Imars, and especially highlighting new features such as the new arena batch, new vintage, and object classification. Before we even start acquiring images, we should consider a few things. First and foremost is to know what the analysis objective is and what we want to extract from the images. It helps to think about what the final figures that you want to put in the manuscript, then use that as a guide to design experiments. By knowing what we need for the final figure, we can then acquire images with sufficient quality and quantity and make the remaining workflow a lot more streamlined. Probably the most important thing to consider is the spatial-temporal resolution that is required for analysis. Think about what is the smallest feature or structure that you are interested in. If you work with time-lapse imaging to capture dynamics, think about how fast the objects can move or change their features, and that should provide a pretty good starting point for deciding the voxel size and time interval for image acquisition. If we oversample, for example, if the objects that we're interested in are in the scale of, say, 10 micrometers, like nucleus, then acquiring the image with 100 nanometer resolution means we'll be working with a much larger image than necessary and utilizing a lot more computational power with probably very little benefits. What happens more frequently, of course, is the issue of undersampling. We have decent resolution on the XY plane most of the time, but the Z step size is much larger, and that can make object detection more difficult with less accurate position estimation and segmentation result. It's also common to find images that do not have enough time resolution for reliable tracking. Sometimes we may hit the spatial temporal resolution limit imposed by the microscope itself, and it's worth looking into the appropriate instrument for the experiments that you are designing. Also, the positive and negative controls for experiments are crucial for image analysis standardization and automation. It would help us understand if we need to normalize across the images and what's the best image processing method for normalization. It will also provide us a guide to the optimal object detection parameters that hopefully differentiate real signals from the background. Eventually, consistency is the key for making the downstream analysis easier. The less variables we are dealing with, the simpler the process. Keep all the spatial temporal resolution the same for one single set of experiments, and if possible, keep all the acquisition settings such as pixel dwell time, laser power, filter sets, and anything that could affect the data acquisition the same, so we can minimize the need to manually adjust individual images. Once the data are acquired from microscopes, Imars file converter can take these raw files and convert them into IMS format that can be opened in Imars efficiently. We support all the major commercial microscope companies' output formats, and if you have a home-built system, we can also convert TIFF series or OME TIFF. The file converter is free, and you can download it from our website. It not only recognizes the image data, but also recognizes the metadata that is saved within the file, so that it will reconstruct the image with the correct XYZ channel and time dimension, and also load the correct voxel size and time step. Of course, not all files come with metadata information, and in some cases we might have to adjust these parameters. In the following demo, I will cover the basics of batch convert files using File Converter, and also how to adjust some of the parameters when the file converter doesn't recognize the image structure correctly. Here I'm going to demo how to use Imars File Converter to convert files into IMS format. In this folder, we have CCI file, which is a file format from Zeiss Microscope. We have a folder called HeLa Cell that contains TIFF series. We also have a folder called Cell Demo No Delimiter, which also is a TIFF series, but it does not come with a delimiter. And I will show you the option to specify how do you reconstruct the file if it does not come with a proper delimiter. Double-click on Imars File Converter to open it. The interface is quite simple. You can also just directly drag and drop the file into input. And as you can see, it will recognize the CCI as input and output as an IMS. Same thing for the TIFF file. Drag and drop. As you can see, even though I only drag and drop one file, it actually recognizes all the 220 files that come with a similar naming, which is HeLa Cell. It will convert into one single IMS with the structure that are recognized based on the delimiter. Now let's click Start All. The new IMS file will be generated in the specified folder. Here, I choose to save it as in the same folder of input file. Now let's look at this special case. What if you don't have the proper delimiter? Again, I'm going to drag and drop into my input. It will still recognize all the TIFF series that have the similar naming. However, if we click on Settings, we see that it recognizes this as a 3 time points 18Z stack TIFF. In reality, it's actually a file of 3 channels. I can simply change the dropdown from T to C to specify it as a channel, whereas the second part, it is Z slices. That's correct. I can also specify the voxel size if it's not recognized in a correct way. This one I can change my Z voxel size to 0.8. Click OK and Start All. Now all the files are converted and we can open them in Imars. Let's open this one as an example. Now this image has been loaded with 3 channels. The display color may not be loaded correctly, especially if it's plain TIFF file. It will not have the instruction on what should be the default color, but we can simply change it by clicking on the channels. Now that we have images converted into IMS format, we can leverage the new batchability that has been introduced since Imars 9.3. The newly designed arena allows us to browse our image data from its original folder. We can also build the batch wizard that contains both image processing and object detection in one single pipeline. It also comes with some handy tools that might be less obvious. For example, we can choose to hide the images that we don't want to be used in batch. We can also undo a batch if we find a mistake in the parameters. There are several useful tutorials available on our website, but I'm gonna cover most of the basics in the demo now. When we open Imars, the first thing that we normally see is the arena view. This is where we can browse image files from anywhere on your computer and organize the files in a way that match your workflow. First, we can configure several folder lists based on either different users, if you're using a core facility computer, or based on different projects. Go to File, Preferences, Arena, or click on this tool icon to bring up the same menu. Here I added a list named Demo. You can add as many as you want. To add a folder into the list, click on Observe Folder and select the folder for including in Arena view. Now all the subfolders will also be included. Here we are going to work on Project 3D folder. In this folder, I have Control and Treatment group that contains 3 images each, and I want to understand how many green objects are within each individual image and compare whether there is difference between the control and treatment. There's also this additional folder called Control Test that I want to exclude from my analysis. We can right-click, remove from Arena, to hide that folder from Arena. You can choose to show or turn off this removed item so that it makes the Arena view cleaner. Now let's go to Control Group and select one of the image as our representative image for setting up batch. Click on New Batch, and the very first step is image processing. From the menu, you can see many functions provided by Imars for image processing that can improve your image quality before the object detection step, including deconvolution. Here I'm going to apply a simple Gaussian filter on the green channel only and also on the red channel. And you can compare from the preview window. The second part of the batch is object detection. You can create as many objects as needed for your project, and you can include multiple types. Here we are going to create a spot. Once it's done, notice that there is this checkbox, Batch stores processed image. Remember in the previous step, we performed Gaussian filter on the image. If this is checked, then that Gaussian filtered image will also be saved. If this one is not checked, then Gaussian filtered image will be used for the object detection step, but will not be saved. Let's click OK to finish. We'll name this Test. So here is our new batch parameters that we can apply to multiple images. We want to apply this batch not just within the control folder, but also to the treatment folder. We can cut and paste it in the upper folder so that when we do right-click Run Batch, we can use the option to include subfolders. On the right-hand side, it shows you the progress of batch jobs. Now both are done. Let's take a look of the folder. Here you notice some differences. First of all, it shows number 2, means that it actually has the original copy and also the copy after image processing. Then we also have the objects that we created, which is a spot in this example. If you click on the image, you will see that the spots are detected and also the images are Gaussian filtered. In the original folder, you can see the original copy as well as processed copy inside the same folder. And there are a few interesting things that you can do with it. First of all, there's restore points that allows you to restore the image back to the original copy. So let's say for some of the images you actually don't want to include the Gaussian filtered results. Then we can just simply click revert and it will revert back to the original image. We can also delete just the objects itself by clicking here, undo the object creation batch job. So now in one single click, we have processed all 6 images within the folder. And of course, if you want, you can move or copy these parameters to other folders where you want to apply the same parameters for batch as well. With all the analysis that we have done, now let's talk about how to export the statistics we generated from the batch object detection. There are a few different ways to export statistics depending on the scope that you want to export. First, we can export it from each individual image file. This option allows you to check and edit the result prior to export. We also have the one-click option to export everything from the batch run. You can choose which statistical values to export from Preferences, Statistics tab, and with one single click, all the statistical values from all the batch files will be exported within the same Excel file sheet like the screenshot on the right. Finally, we have the option to export statistics from Vantage. In our latest release, Imars 9.6, there are several nice new features in Vantage that I would like to share with you in our following demo. The advantage of using Vantage is that it gives you a preview statistics in graphs form, and you can directly have publication quality graphs or plots from Vantage without the need to export to Excel. But even for exporting statistical values, the export format from Vantage sometimes can be quite helpful. We'll go through all these statistics export options in the demo. After running batch on control and treatment groups, let's take a look at different options to export statistical values. You can of course go into each individual image and check the results. You can even edit spots, for example, if you see that there are spots missing, or you might want to delete some of the spots that are falsely detected. And then you can go to Statistics tab to export the values of interest. This, of course, might not be very feasible if you have many data sets. Alternatively, you can use the one-click option on the right. This is going to export all the statistical values that we run within this folder. You can configure the statistical options to export by going into Preferences, Statistics, and configure the ones that you want to include. The third option is to export the statistics from Vantage. Let's say we not just want to compare the number of objects that we detected, but also we want to compare, for example, the intensity value of these two groups. I can highlight both folders and click on New Plot. This will bring us to the Vantage Plot, and we have 3 different options. 1D view, which allows us to compare one single variable, 2D view for 2 variables, and objects view for up to 5 variables. Here it already grouped the data by its folder. So this one is from the control group and this one is from the treatment group. I can choose the plot type to match the ones that I'm interested in. So let's say I want to compare the intensity mean of channel 1. At the bottom, you can see the summary of these 2 groups and also the detailed view of individual objects as well as which image it comes from. I can save this entire table. and call it scatter plot or any name that you want. Let's take a look at the exported format from all these three options. First, let's take a look at the single image option. You can see all the statistical values are saved as independent Excel file. We can take a look at the overall file where it will tell you the total number of objects being detected. So here we have 58 objects. The batch statistics export looks very similar to the single file one, but if we look at the overall option, it tells you not just the number of objects, but also which file it belongs to. So in this particular case, we have 3 files from the control group. That's why we have 3 different numbers, and each of them linked to individual files. The scatter plot, which is saved from the Vantage, is in a slightly different format. It organizes the data based on the variables that you choose. So here we only have one single variable, but if you choose the 2D view or the object view where you can save up to 5 different variables, line up row by row. And on the right, we have where the image this is linked to, as well as the object ID, so that you can go back to that particular file and find the specific object for that particular value. So this is another way of organizing the data, and really depending on what is your batch analysis goal, you can choose one of the options that we offer for the export. Now that we have covered all the basics, including converting the files into IMS format, organizing the data in Arena, setting up the batch parameter, and exporting statistics, now I'll do 2 more advanced examples of batch processing to showcase other features in Imars that are hopefully useful for your projects as well. First example, I'm going to use exactly the same data set as before, but asking slightly different question here. I want to know how many red spots are within the green surfaces. This is a pretty typical colocalization questions that we encounter a lot, and we can actually address it using batch as well. So just like before, let's select one of the representative image from control and start a new batch. This time I'm going to skip image processing step, directly go to next. Now, for performing the colocalization analysis, one key is to understand which objects you want to create first, because the shortest distance calculation that we are going to utilize later can only be performed on the existing objects. So for this example, I'm going to create surfaces to find out the green signal as my first step. I will keep the shortest distance calculation option. And get rid of something that is way too small. Now let's create spots to identify the red spots. I'm going to turn off the surface detection so that we can focus on the spot itself. This time I'm going to keep both classify spots and shortest distance calculation in my option. Source channel change to channel 2. Here under the classification step, we can change the name and rename the set to green location. We can utilize the shortest distance calculation to classify the spots that are inside or outside the green objects. You can zoom in into the histogram if you want to make it more precise. I'm going to find a number that is as close to 0 as possible. You can also change the class name right here. So the blue, I'm going to call it inside. And the purple represents outside. Go to next. Rename my spot to red. And we're done. Again, I want to apply this to both control and treatment group. Now I will run batch including subfolders. So for the control group, now everything is done. We can see both surfaces and spots are created. On the right hand side, let's take a look what Vantage Plot looks like. Click on Vantage. Here it listed both the spots and the surfaces. We can turn off the surfaces and focus on the spots. You can see the aggregated behavior. Let's change this to, for example, we want to understand whether there's an intensity difference. And immediately we can compare the spots inside and outside the green objects. We can either plot it together or separately. This new classification also is saved in the table. So when we export the statistics from Vantage, the information whether the spots are considered inside or outside will also be saved, in addition to the parameters that we are interested in. So this is a very convenient feature where you can add as many classifications as you like so that you can group the data in different ways and still manage to look at them in an aggregated feature. In this second example, I want to share with you a time-lapse data set. Let's take a look at one of the example. Here you can see a lot of spots are moving around. And we want to ask two questions. First one, whether the intensity is associated with the spots aggregation level, which we can now measure using the average distance to 3 nearest neighbors statistical value. And the second one is whether the average speed of movement is associated with intensity level. So let's see how we can do that with batch. Let's go back to Arena and start a new batch. I will also skip image processing for now and create a spot detection. Here we will do the track spots over time so that we can get the speed statistics. We will also classify spots so we can categorize them based on the aggregation level and also based on the speed of the track. We don't need shortest distance calculation for this example because we're only looking at one single population. Let's go to next step. Now we might want to only select the ones with longer duration so that the statistical values are more reliable. Okay, under the classification step, let's set the first one as aggregation. The input type will keep it as all spots. And the aggregation will be using average distance to 3 nearest neighbor. Let's find a cutoff that we feel reasonable. So let's say we put 7 micron as our cutoff. We can add another classification, but before that, let's change the name. Let's add another set. This one is speed. And instead of looking at all spots as our input, we're going to look at the track as our input. We'll choose track speed mean as our classification filter. Here I'm going to put 1 micron as the cutoff. Now, both of the classifications will be recorded. Click OK. Running the batch on all 3 images. Depending on the data set size, of course, you might expect the batch run to be a little bit longer. Once it's done, let's take a look of individual images here. Go to Spots, Statistics, and here you can directly see how many spots belong to each individual classes, as well as how many tracks belong to the different classes that we specified. We can also look at it from the Vantage Plot. Now, from the 1D view, let's first take a look of the distribution of the spots itself. Then because we separated using the average distance to 3 nearest neighbors, you can see that the plot right now has a very clear cutoff. But what we're more interested is whether it's related to the intensity itself. So let's change this to intensity mean. And as you can see, it actually is pretty similar. We also provide the statistical test directly if you want to look at the p-values as well as more statistically related results. We can also compare the speed and turn off the spots related ones. And because these are generated with the tracks, you won't be able to see them unless you change the plot type to the track specific statistics. Here we're going to look at track intensity mean instead. And again, on the track level, we also don't see the intensity difference based on the speed classification. But here it should show you a pretty useful way of looking at the data at spot level and also at the track level, and you will be able to plot them from the 1D view, 2D view, or even the objects view. Just notice that the objects view would take a lot more computational resources. So if you have many objects, using 1D view or 2D view will give you faster way of visualization. Hopefully today's webinar provides an overview of how you can analyze the data efficiently in Imars using our batch process. And if you have more questions, feel free to contact us at ussupport@bitplane.com. You can also find more resources at our website. Thank you for attending.
Thank you so much, Julie. That was great. And we have gotten a few questions. We need to transfer over what we're showing. So just give us a second with that. Okay. So we had a few questions come in, not too many, but it would be good to ask these out for the whole audience. So, Chile, one person asked, should you determine the parameters for spot detection for, and this would be true for other types of detection too, should you determine those parameters for control and experimental images separately, or should you have one spot detection parameter for all conditions? And you know, you were in the past a bench scientist as well, so you can probably speak from experience and outside of being on the Imars team too. But yeah, could you talk a little bit about that for us?
Sure. Hi, Meredith. Hi, everyone. Can you hear me?
Yeah.
Okay. And so the short answer is that we do want to keep as many parameters consistent as possible. And that's why the batch is very powerful in that you use one single set of parameters to process all the images from the same experiments. But of course, there are cases where perhaps this, for example, the cell size in the control group and in the treatment groups are so different. And what you are more interested in is the, for example, counting the number of cells rather than determining the size of the cells. Then in that case, you might want to change your parameter based on the image so that you can capture the quantitation that you're looking for more accurately. But in general, we do want to keep our parameters as consistent as possible.
Yep, good, perfect answer. And if you have questions about what you should be doing or how to handle a specific situation in one of your images, feel free to reach out to the support team. Chile put in the email address for the US support team. If you're in other regions of the world, you can email them. They'll get you to the right place. We have teams all around the world so that we've got someone in your time zone. But yeah, we can help you with specific things as well. So another question that came in was, can you please explain a bit more what the Gaussian filter is actually, if it's actually modifying the images? And you know, this is a general Imars question. It's not so specific to batch, but I think it is a good thing just to reiterate to people what that Gaussian filter during the creation wizard is doing. Yeah.
Sure, yeah. So here you can see the example that we just showcased previously, where we have the Gaussian filter applied. Let me open this image up. And what you can see is that the image looks a lot smoother with less sharp features. The original image looks like this, where you can see a lot more intensity variations. So what the Gaussian filter is doing is basically smoothing out your image based on the filter width. And you can think about the filter width as what you want to preserve as the smallest feature in the image. So in the case where we just discussed previously, what we want to detect is the much larger feature, for example, here. Whereas the variations that we see in the original image is at a much smaller scale. And that's why applying a Gaussian filter where we can smooth out all these smaller variations can help us with the object detection better, especially if what we're trying to detect is much larger than the original image resolution.
Mm-hmm. Great, thank you. Okay, and then a question sort of spinning off from that. So we're seeing you as part of a batch, as a batch workflow. You're using the Gaussian filter here. Someone asked if they can make the display adjustments that they're often using. If you pull up the display adjustment window, they're asking if they can batch process those.
So unfortunately, the display adjustment itself is not batchable. However, whatever you do within the display adjustment affects only the image visualization. It does not affect your data quantification. And our current batch pipeline is based on the quantification need. And that's why the display adjustment is currently not part of it, but it is certainly something we'll consider in the future. Thank you.
Yep. Yeah, it is, definitely. Okay, so I think we've had a few more questions roll in, but I'm going to save those just to, for Daniel to maybe answer, and then I'll get those out to the rest of the support teams and support team members to answer for everyone that's joined us today. So if you do have questions, you've got a couple more seconds to get those in. If not, you have my email address. Again, I'm Meredith. I've been the one sending you lots of emails lately about the homeschool, and you can email me and I'll get you to the right place. Let's see. The other thing I wanted to say is that we're going to take a little bit of a break. We have realized everyone and their brother is on vacation right now, so we're going to take a little bit of a break from the homeschool series before we wrap it up with our last round. So just stay tuned for an email from us where we're going to email out the next set of webinars that we'll have and the dates for those. So we're going to keep it going a little bit longer, but it'll just take us a few weeks to get back into the swing of things again after some summer vacations and staycations. But thank you so much to everyone that joined us today. Chile, you did a great job. Nice and clean and easy to understand. I really appreciate it. And yeah, thanks to everyone for tuning in, and enjoy the rest of your day, the rest of your week, and take care. Bye-bye.